Right, this is the start of B3 and this topic is called living and growing, and so what we're going to be doing is basically looking really closely at how that procedure actually takes place. Now for this first part of the topic we are going to be looking at some new organelles in the cell that you probably may not have heard of before, and also looking at a little bit more detail at DNA. Now, as you know, cells are the building blocks of living organisms. Cheek cells are typical animal cells, and each is enclosed in a membrane which contains the cytoplasm and organelles such as mitochondria, which I'll talk about in a minute, and a nucleus. Inside the nucleus is the DNA that carries the organism's genetic code. So if you look at this diagram on the left, you will see this is a diagram of a cell, and if you look closely you'll see that dark patch is the nucleus. If you looked inside the nucleus you would see the chromosomes, and then if you look closely at the chromosomes are made of coils of DNA, and it's called a double helix. Each section of DNA is a gene, and this is a code for a protein, and I'm sure you've heard that proteins are for growth and repair. Well, there's a lot more to them than that, and we're going to be having a look at how they're made and what they actually do. So if we look at DNA more closely, you'll see that it's made up of four bases or pairs of chemicals. You have A and T, so you've got adenine and thymine, and C and G, which is cytosine and guanine. As far as I know, you haven't got to learn the names, but you do need to know that A always goes with T and C always goes with G. And if you look at this diagram of the DNA, you'll see that you can see like the blue and the reds are always together. They might be the other way around, but they're always together, and the oranges and yellows are always together. So that's trying to show you A goes with T and C goes with G. Now the people who actually are accredited with finding out about DNA are Watson and Crick. In 1953, Watson and Crick were at Cambridge University in the UK, and they used data from work of other scientists to work out the structure of DNA. Rosalind Franklin at King's College in London obtained X-ray data which showed that a DNA molecule consisted of two chains wound into something called the double helix. It's a bit like if you look at a ladder and then twist it, that's what it looks like. Watson and Crick used her data and help from chemist colleagues to build their model of DNA, and it's like I said, it's called a double helix with pairs of bases forming cross links. They published their paper before Franklin, and so they got all the credit. In 1962 they shared the Nobel Prize with Maurice Wilkins, who was Franklin's supervisor. Franklin died aged 37 in 1958, and the Nobel Prize is not given after someone's died. So she was never actually accredited for the work that she did. If we look at two other structures that you're going to come across in the cell, first of all you've got this thing called the mitochondria, and if you looked inside a cell it basically looks like a little sausage, but really this is like, without this you just wouldn't be here, because this is where respiration takes place. So when your glucose is taken in through your food and you absorb it into through your intestines and it goes into your cells, that glucose has to go into the mitochondria, and then the oxygen that you take in through breathing goes and attaches to your red blood cells, then it is delivered through the capillaries and it goes into the surrounding cells, and again that oxygen has to go in and go into the mitochondria. And that's, like I say, where respiration takes place. So you need to know the equation for respiration. So it's glucose plus oxygen gives you carbon dioxide plus water plus energy. The other thing you need to know is this little thing called a ribosome. Now ribosomes are really tiny, and they help to make proteins from something called mRNA. holds the mRNA and the tRNA in the right position for amino acids to join together to make a protein. And we're going to look at this process in a little bit more detail in the next video.